Good morning, everyone. I am Chris Rebelo, the Senior Director of Communications here at the Buck Institute. Happy New Year and welcome to today's event. I hope you all had a really wonderful holiday. Today, we are going to be talking about a mechanism that is involved in aging biology. It's a mechanism that, when it goes awry, has been linked to a number of age-related diseases. That mechanism acts as sort of a housekeeper in our cells, which gives me the perfect segue to my usual comments about housekeeping here in our auditorium. So please, if you haven't put your phones on silent, please do so. And also, I always point out the emergency exits to keep the fire marshal happy. So, who's new to the Buck? Anybody starting off their new year? Welcome, welcome. We love to have newcomers. Rather than me going on about how great our scientists are, I want you to hear from them. We have a very short video. Joran, if you could play it, that would be great.
The Buck Institute is interested in understanding aging, not only to make us live longer, but to actually alleviate and suppress the development of diseases.
Chronic disease is the great health challenge of our time. Aging is the main driver of chronic disease. To eliminate chronic disease, we must harness the biology of aging so we can live healthy longer. The Buck Institute is the world's first research organization dedicated solely to the biology of aging. We work with the building blocks of biology to understand how we age.
The Buck is concentrating on really trying to go to the root of the problem, so understanding that we can sort of intervene really early.
The Buck Institute is probably the best place in the world to be working on aging because there's so many different aspects of aging that are being studied. It's key because no one lab can do it all.
Our scientific explorers are brilliant, curious, and committed.
The idea that you could actually develop drugs that slow down the aging process was really something that 20 or 30 years ago people thought this cannot be done. Not only can it be done, but we will do it.
The pieces are in place. And we are in a unique position to put them together. Our world is defined by paradigm shifts, created by scientific discoveries. The biology of aging is the next frontier.
We're working toward a world where healthcare is better. It's a world where if we can eradicate the aging component to disease, we're all going to live healthier and longer.
It's just an incredible vision that really will, surpass anything else that we've ever seen before.
We are inviting you to a future of aging without illness. It's happening today, and it's happening here. Join us.
Great. So before I introduce today's speaker, I want to thank our sponsors. They are listed in your program. I also want to make sure that you all have note cards that you can write questions on. If you have a question, raise your hand. One of our volunteers will pick up those cards and bring them up to me. If you want more cards during the event, raise your hand. Our volunteers will get to you and make sure you get taken care of. As I look back to the back of the auditorium, I want to welcome those of you who are on Zoom and remind you that you get to ask questions too. Just enter them in the Q&A section on Zoom. Somebody will get those questions up to me here as well. So, it is now my pleasure to introduce Dr. Malene Hansen. Malene is our Chief Scientific Officer here at the Buck. She's also internationally recognized for her expertise on autophagy, especially as it relates to aging. And we're going to be talking about autophagy today. You can read about Malene's background, her educational and professional background in your program. Know that she hails from Denmark. She got excited about research on aging during her graduate studies. She is a molecular biologist who has received a number of prestigious awards for her work. Malene, come on up. We have a new clicker for the new year. I hope it works. Okay, here we go. Yes, okay, autophagy, right there in the center of the screen. What the heck is it?
Yes, let's talk about that. Very excited to be here. Happy New Year to everyone. And thank you for inviting me to chat about my favorite topic. So autophagy is another word actually for self-eating. That sounds a little funky, but it's basically equivalent to when you recycle at home. I assume most people here do. So we use an item like the Coke bottle here, throw it in the recycle bin. There is a truck that comes pick it up, and it sends it to the recycle center. Our cells, everybody's cells in their body is doing this all the time. Probably especially a little bit more right now because eventually you're going to be a little hungry before lunch. We'll talk about that. That's a trigger for some of this. But inside in this ring, it's sort of the equivalent to what happens in each of our cells. So the cells have an ability and a need to clean up, just like we do in our houses. So the depiction here is a mitochondria. That's sort of the powerhouse of the cell, if you wish. They can go bad. That's not good for the cell. They need to take it away. They put it into trash bins. It's essentially like a bag. It's a little vesicle you can see here. It has basically like a trash bag around it. And that trash bag can actually travel inside the cell. It's a little bit like highways, if you wish. They're little motors that basically move vesicles around. It literally looks like shoes that are sort of walking around what's called microtubules, but you can think of it as the highway of the cell. And it moves them into other vesicles that actually keeps these little Pac-Mans in them, as you can see. And that's the equivalent to basically chewing up that material that you want to get rid of. And so all the little building blocks that come out of that can then be used in the cell to build not a new Coke bottle, but a new mitochondria or like a new lipid droplet or any of the other things that the cell really needs to have. So it's kind of a gatekeeper to keeping everything neat and clean, especially in stress. So we can talk more about that.
So I got so thrown off by having a new clicker, I forgot to remind everybody that Malene is a PhD. She's not an MD. So we can't give medical advice here. Anything we do talk about today, please talk to your doctor before you make any lifestyle changes. And also, this event is being recorded, and you're going to get a follow-up email that will contain a link to today's event. So you don't need to take notes. Just hang out and enjoy it. And if everybody can hear us okay, right?
Great.
So if there's a problem with hearing, raise your hand and we'll get somebody to boost the sound. Okay, so I think the cool thing about autophagy is that it really can help the body create new things.
Right, from old and damaged things.
But the bad news is that it tends to decline with age, right?
Correct. And you can also imagine if you do too much of it, you can overeat, and that's not good for the cell either. But the problem with the aging is, yes, that it tends to, like many other processes in our cells, which is very much what the labs here at Buck are interested in studying, is how does it kind of tend to fall apart? With the idea that if we find that out, there may be ways that we can come back in and fix it. Kind of like you need to know how a bike works before you can mend it when it breaks down. We don't quite know exactly how it breaks down, but we're starting to learn that. So there are indications in different systems and cell types that, for instance, you can run out of these trash bags So of course, if there's nothing, if you can't put the trash in something, it's not going to go through the whole cycle. There can be holes in those highways, so like basically potholes, so that the transport between the cycle, the recycle plant and where you basically deposit the trash is not going to work. That would also sort of basically destroy the whole flow of things. So because it's such a linked system, there are many ways to sort of break it. And so obviously we're even more interested in finding if there's something even sort of further upstream that can put the whole thing in place and make it run better for longer. But each way it can kind of break can be a way for us to potentially fix it again if we know how it happens. So that's what we do. The lab here at Buck, and they're all here, are interested in, it's very, obviously very microscopic work, but figuring out exactly how this process is put together and how does it do what it does when it does things well. That's the segue to the top here. So autophagy is really important in a number of things that it does on a normal basis. So for instance, when a cell and an egg get fertilized, like with sperm or any later developmental programs are often affected by this in terms of creating nutrients and other ways to basically run the right way. Survival under stress, you can imagine how if you have a system that can sort of bring things and clean it up, if you stress the cells, like you have a room that's really untidy, you want to kind of keep it clean. So this process can kind of help keep everything in check like that. Quality control of proteins, you're probably well aware that many of the proteins are sort of the building blocks of our cells. They actually can tend to clump, especially in older age. So neurodegenerative diseases like Alzheimer's disease have a sort of a culprit for proteins that start to clump together. They can interfere in that way with the functions of the cells. And again, you can imagine how you can build a trash bag around one of those clumps and send it away so the rest of the cells doesn't see all these problems anymore. So it's really good at that. And then it can defend against pathogens. And some people may argue that's probably one of its really original roles in the, like a really ancient time where cells lived alone and in smaller organisms and could get infected. And so you can again imagine how the cell could build a trash bag around like a pathogen and send it away before it starts to do its bad thing within the cell.
So we have some examples here. What happens when autophagy goes off the rails and is dysfunctional?
Right. So things can break apart. The cycle I just kind of put in that looked very simple. It's obviously much more complicated than that. So there are many different proteins or enzymes within the cell that's important for building this trash bin or putting together like the motors that moves it, or the enzymes, the pacments that have to basically degrade the material that comes in. So many different genes, actually more than 40 genes, of which many have been linked to disease itself. And so you can imagine, I already touched on that, neurodegeneration. If you cannot fix and take away some of these clumps, that can actually be a real a problem. So there's a number of links to neurodegeneration from autophagy not working. Again, we don't know exactly where it stopped working. So if, again, we can fix that, we would perhaps have a way to especially help with neurodegeneration. Cancer is much more complicated, but there are many links between cancer and autophagy. You can imagine that a cancer which wants to grow progressively could take advantage of this process and generate even more nutrients out of this kind of cell cycling process. So there are some cancers that do that. Obviously, in those cases, you would like to inhibit autophagy.
And we're going to talk about cancer a little bit in a few minutes when we get to some interventions that impact autophagy.
But many links there also oppositely where, cancers can inhibit autophagy. So same for heart disease. You can imagine that especially a cardiac muscle that is running a lot of energy, it uses a lot of these mitochondria I already showed you that can go bad. And so especially a heart cell would like to keep all of its mitochondria really healthy. And so if that doesn't work, you can also end up with heart disease. Or directly relate to aging, which is what our lab studies and many other lab studies, in terms of direct links for that. And like the video showed, right, like aging is the biggest risk factor for not only these, but many other diseases. And so we like to think that studying autophagy as a common potential mechanism or driver would help us perhaps get towards future therapies. Not what our lab would do. We try to figure out how the bike is working, and we will talk to the bike repair people and see if they can help us with the rest.
So we have somebody in the audience who's into biology, which is great. Please clarify, where are those little trash bags disposed of in the body? What happens to them?
Yeah, excellent question. So first of all, every cell, so whether it's in the liver, in the heart, in the brain, can do this. Every cell needs to keep itself clean. So the bag itself actually gets burned up, if you wish, so in this recycle plant. So it's called an intracellular process. You keep your own cell clean. But the question is actually even better in that regard, that newer investigations that we're actively also working on is that there is actually some cells that seem to not even bother to send it to their own recycle plant, but they throw it out of the cell. And so, other cells can kind of help take care of it.
So you throw it out the window and you hope that somebody else takes care of it,
cell
basically.
that can take care of it. So we know very little about that. That's a relatively sort of new angle, this secretory autophagy, if you wish. And so that's something we're really excited about. But mostly when I talk about autophagy, it's cleaning up your own cell. And so each cell within an organ and ultimately in the organism will be able to do that.
So a chicken and egg question. Does autophagy go bad and then we age? Or do we age and then autophagy goes off the rails?
Fantastic question. I don't know. That's really the essence of a lot of what we do. We can often do, and we'll talk about this too, that we can look at correlation, right? So sort of things that move together. But that doesn't mean one thing costs the other. And so I don't have an answer to that question. But hopefully we'll figure it out.
Okay, that's great. I don't know is a great answer in science because it gives us something to work on. All right, so everything in your lab here, a lot of stuff here at the Buck starts with basic research. Let's talk about some of these model organisms. Why are they so essential to the work that you do when you look at these cells?
Right. Yes, so absolutely powerhouses in terms of finding new ideas and getting basically a handle on how to approach this. A yeast cell is a single cell, it's not an organism. So the smallest and simplest multicellular organism, meaning with different types of organs, a worm that we study, they live they're very tiny. It's a comma in a book, but it has almost 1,000 cells. We know exactly who they are, where they sit, what they kind of do. We can look through it under a microscope. Obviously, they're too small to just look at. And so we can learn a lot about each cell and what they kind of do. A fly, it's a little more complex. For instance, a fly has a heart. The worm would not have a heart. So, imaging or studying heart disease in a worm may be, more of a limitation. So there are limitations, but opportunities in all of them. And then, of course, a mouse is much closer to us in terms of it has more behaviors, right? Like it has hearing and other things that we would like to learn and understand about. But what's common for all of them, relatively speaking, is that they age really fast. So for our research, that's a really important factor. A worm lived for like a couple of weeks. So you can see we can do many, many experiments. A fly, this is a typical, fruit fly. It lives typically for a couple of months. And then the mouse would live probably about a couple of years if it's in a laboratory. Obviously in the wild, it may live shorter because it gets cold or have other sort of threats for it. But the real key is rapid aging. We know the genomes for all of this, which means that we know all the players like all basically those building blocks that, becomes proteins and enzymes that needs to do all this job of metabolism or autophagy for that matter. And then we can do experiments with them, meaning we know pretty well in every one of these how we turn genes up or we turn them down. So we can start asking, does it really matter if we have the activity of this gene when we want to look, for instance, on aging? So the underlying purpose is that they're all similar in some ways. Like we all eat lipids and carbs and, fatty acid. They look a little different, right? I mean, we eat different things. A worm would eat bacteria, like on this plate. We like to eat, steaks and potatoes or whatever. But they're essentially the same building blocks. So we have many of the same enzymes that have to take care of it. And so that's the sort of premise for using them and hoping that we can learn something relevant to us. In fact, if we see something that's true in all of these, like autophagy being regulated with age, we like to think that probably, not for sure, but probably is also true in humans, right? Because we would then be able to deduct that it's an old program, like it's an ancient program that, is something that is fundamental probably for function in whatever context it may be, reactions to stress or something like that.
So here we get to interventions that can modulate autophagy and recycling. So I'm going to start with dietary restriction. And I'm going to ask you, because I know your lab, you have a lot of projects, but a number of them focus on dietary restriction. What are you doing in your lab?
Yes, so we do a lot of things, and many of them are actually sitting up here. So credit to them, they do a fantastic job figuring out cell biology-wise how this really intricate process may play into aging and age-related diseases. There are diseases we also can model in these C. elegans worms. So dietary restriction is one of the best known ways to actually extend lifespan across organisms. You eat a little bit less. Obviously, you don't starve because that would be malnutrition. But what the point here, and actually to the side of discussing this, I think it's important to discuss the concept of causality versus correlation, because many of the data that we're going to talk about and that is linking autophagy either to human health or to health in general, also in these model organisms, are done in sort of two categories of experiments. And I think it's important to sort of think about what each of them mean. So under causality to my left, is actually an experiment I did as a postdoc many years ago at UCSF. And what it shows is a diagram of basically looking at how many animals are alive at a given time. So there's a fraction of worms alive. Typically, we look at about 100 worms to the left. And then you can see days on the bottom line. Is the time of life, and that's the kicker I just told you about. It goes really fast, right? So what you can see is that there are normal fed animals. That's the black line. Some start to die early, some die a little later. That's actually how the human lifespan curve would look like, right? Some people would die earlier than others. So you get a curve, and you talk about a mean lifespan. It's sort of when half of them are still alive. Then you can dietary restrict them. In worms, you can put a little less bacteria on there, or we have other tricks to not have it eat as well. And you can see they live a whole lot longer. They start dying later, and that curve comes out much further, the blue line. That's what's called lifespan extension. These worms live longer. We're really interested and important to note. We also, of course, are interested in having them be healthier for longer. So they swim better. There's a number of things they do better. They can smell better. So that's all a good thing. Dietary restriction works very well in worms that way. But then, and this is the causality point that I wanted to make, we can do an experiment that can be really hard to do in other organisms, or at least the more complex ones. And that is we can use genetic mutants. So basically an animal where we have tweaked the autophagy process down, meaning we take one of the genes out that is making that trash bin, for instance, or making the trash bag, so it stops the process. That's really important. We can basically engineer the process to stop. And if we dietary restrict that one, that's the dashed line, you can see the dietary restriction doesn't work anymore. That's a super important result because it's a direct piece of evidence that autophagy is required, a driver for the dietary restriction effect on aging, not just correlation. And so that's what I have on the other side. I think correlation in this way, this idea that two things move together, but one doesn't necessarily cause the other. In the summer, you sell more ice cream and you also get more sunburns. Those two things correlate, they move together, but there's absolutely no evidence that ice cream should cause sunburns. The real cause is that the sun comes out, right? And that makes the two things move together. So that's an important distinction, that we can do causality, like I showed you the experiment we could do in a worm. For most purposes, at least in the mice and the humans that are in this little diagram, in dietary restricted animals like mice or in humans, you see long lifespan or better biomarkers in humans. Humans are not a great lifespan model because they live so long, right? Like it's almost impossible to do the full experiment. It's been done in primates, I should mention. They live maybe about 30 years. That's also a really long experiment. So it has effects in at least some species on lifespan, or these better biomarkers, like for humans, better lipid profiles, insulin resistance, things like this. And then you also see autophagy increase. But in those cases, I don't know if one leads to the other. They just come together, right? So that's an important limitation for pretty much actually all of the human studies that we're going to talk about, that we just see things move together. Doesn't mean that they cause each other, but there's pretty good evidence, and at least from yeast and worms and flies, where we can do the trick, like I mentioned for the worms, where we can have a mutant that can deliberately take out the player we're trying to ask about. Otherwise, the lines of evidence are correlative in that regard.
So I used to be a reporter way back in the day, so this is my big takeaway that I want you guys to have from today. When you read a newspaper article and it's about some discovery in health, if it has the word XYZ has been linked to XYZ, that is correlation. It's an observation. Doesn't mean that something causes something else. And if there's an article that says scientists have discovered that X causes Y, that's the one to really pay attention to, because they've really drilled down to find out what happens. So I like it when people are smarter consumers of health information. Right. So there you go.
But the pitch is still, right? Like yes. Since it is causative in a number of organisms, there is a likelihood that it could also be causative in humans. It's just extremely difficult to kind of prove that. So we're talking about evidence. All of this evidence is good evidence. Like it's useful, but direct evidence, is a stronger piece of evidence, if you wish.
So I've gotten a couple questions. Does diet affect autophagy? And please explain dietary restriction, eat less and or better. So what do we know about diet and autophagy?
Right. So let me clarify. Using the word dietary is kind of on purpose. Historically, you would also see it often be called caloric restriction. So obviously, there is sort of the point to it that there is the amount of calories, and then there's the quality of the calories. And there's been a number of studies, at least in, again, these simpler organisms, that it's the diet, the quality of the diet, and not just like the calorie amount. Now, exactly what that means in humans, I'm not sure. There's good evidence that a lot of proteins can be good for a number of things, and absolutely studies that show that certain types of the building blocks that are in all proteins, right? Like, for instance, something called proline or alanine, and you can have some types of food that have more of one than the other. So all of that is kind of being worked out for optimization. It's a difficult experiment, long experiment to do in humans. So what a lot of there are trials going on that are more sort of retroactively asking, what do each of the people in the study eat and then you measure afterwards, and you try to again make a correlation for the people that have had a lot of protein because they eat steak every day or whatever. They have a certain level of autophagy. It's complicated because the process itself is cycling also during the day, so it kind of matters when you measure. Like if you measure one human in the morning and one human in the afternoon, they also may be different for those reasons, or they have underlying diseases. The environments are much more complicated. So humans are difficult in that regard.
How many here are familiar with intermittent fasting? Have you heard about it? Great. So I got a question. Does intermittent fasting contribute to autophagy?
Yes. And a very prominent one that actually has been studied especially well in flies. And so basically what it means that you eat for a portion of the day, and you may eat the same amount of calories. So that's again this dietary versus calorie thing, but you eat it in a kind of restricted amount of time. For instance, you only eat for 10 hours a day. And then obviously you fast for the other 14. And so there are causative, direct evidence for flies that this intermittent fasting, which extends their lifespans and have other benefits, induces autophagy. And again, if you do the same experiment in a mutant, it doesn't work as well. We're going to talk a little more about that. It has also a lot to do with circadian rhythms. So there is a layer of when do different proteins work during the day. We fast at night, right? So sleep would automatically link to fasting in that regard.
Okay, so let's talk about pharmacology. I want to point out the big line here. No causal mechanisms of action have been discovered yet in humans. So these are pictures of all the things that you can either get at the pharmacy or even at the health food store. We're not going to talk in detail about all of them. I have picked two, spermidine and urolithin A, because they're kind of both hot topics these days. So the first one is spermidine. Lots of interest in this in the field. Well, why is it so hot? What's so exciting possibly exciting about spermidine?
So it's one of the best studied, I would say, in terms of having been looked at in most of these organisms, like a yeast or a worm and a fly and a mouse, and it seems to be working similarly. It would extend lifespan and certain health aspects in an autophagy-dependent fashion. There are additional studies in mice for outputs of like cognition. So cleaning up your brain obviously is a very important thing as well. And so you can sort of pick down all the different health spans or health outlines that you're interested in. And I think spermidine is the one that has been tested in the most depth in that regard. All looking pretty positive in that regard.
So the good news is you can actually get spermidine from your diet. Here are some ways to get it. I do a lot of these seminars, and one thing that I always come away with is that supplements, you just don't know. You know, whether they're going to work for you, whether they're good for you. In some instances, it's good to boost things like autophagy, but as in cancer, maybe you don't want to boost autophagy, right? So that's, how do you approach that, Malene?
Yeah, I would just add here, because there has been, the human side of this is another correlative study where sort of in hindsight, people that had been basically followed for dietary intake and also basically measuring the blood for how much spermidine. It's one of many other metabolites and compounds that you could measure, that there is a correlation with higher level of spermidine and then better health outcomes. Now, those humans could have many other things going for them, but that's at least one correlative kind of observation in favor of spermidine also potentially having, positive effects in humans. The part of when is it good and when is it bad? That's a real challenge for any, of course, intervention, and what works for you may not work for me, which is why we need healthcare providers involved, dietitians, and so forth. But in this case, at least for spermidine, and the concentration it's been looked at, it looks pretty promising. I should say, though, that for anything, you can overeat, right? And so at some point, something is going to get toxic. It doesn't matter what you eat, really. Like you can get uncomfortable with that. So for all of these, and in any context of a new compound, you would do sort of dose response curve or toxicity curves are really important pieces before one moves forward with things like this.
So I didn't put a circle around this one, but I want you to talk about it. I understand that rapamycin, which is a drug, so you have to get a prescription for it, can inhibit mTOR and encourage autophagy. So I have issues with rapamycin. What should people be aware of if they want to fool around with rapamycin?
Yeah, I wouldn't fool around with rapamycin. I figure I would do this in a very targeted way. Step back. Rapamycin was developed as a drug to be given to humans for immunosuppressive purposes, like when you receive a transplant. There's a reason why you may want to suppress your immune system so you don't react to, the organ you may have been lucky to and needing from somebody else. So it was already available, and that's sort of the good news about that particular one is that it's known to work well, right? You can give it to humans, and it's safe. Now that's not necessarily what you want to do, right? Like you don't want to immune suppress yourself all the time. So. But the good news, and so the interest, and correctly stated, mTOR is an energy sensor, an enzyme that sort of helps to monitor what your status of the cell is, and a major regulator of autophagy. That has been linked to lifespan. Actually, Pankaj Kapahi, who's here, showed it first in flies, that you can inhibit this enzyme and it would extend lifespan. So with the drug, you can do the same. It's key that the concentrations that seem to be working, at least in many of these models, are much lower than what you have to give to a patient for this immune suppressive effect. So that's kind of good news if you don't want to mess too much with that side. And that's essentially the problem with everything. There could be a side effect that you're not interested in basically having. So it's promising from rapamycin in that regard, and probably the best studied mechanistically because it hits exactly an enzyme we know very well.
So I know we talked about dietary restriction. I have gotten a number of questions. People really want to know about how long should I fast? Should I fast how many hours a day? Do I fast twice a week? Do we know about fasting and particular how it impacts autophagy and how much we should do? Where's the science?
I would say we have very good news for the worms in the audience and the flies in the audience, but humans is more difficult. So I figured a question like this would come, and I looked it up a little bit. So there's a number of groups that's interested in this, and the best one I can think of that I recently heard give an update on their ongoing trials that uses measurables. Again, you look at a number of people, see what do they actually eat and what works sort of best for them, right? So you have to have a lot of people because there are many other factors that can play in. But that study, which is running in San Diego at UCSD and at Salk with Sachin Pachin would argue that what seems most optimal at this time is that if you can compress to eat, and this is just for health, different health outputs. We don't know about lifespan. Would be a 10-hour window, which means you have to, not eat for 14. But maybe you can do 12 and 12. You still get effects. So the good news is that any level of period would change some of these markers, maybe less so. But I would caution it by saying this is very much research that's ongoing. And maybe something works better for you than for others, right? So again, I would talk to a dietitian and your healthcare provider to see what may work for you, because maybe you have a schedule. For me, I like to exercise in the morning. It doesn't work for me to not eat. It's easiest to do it after you've slept, right? That doesn't work for me. So you have to sort of work it out with other social factors and so forth. But it's an excellent question, obviously, that people are very interested in.
So let's talk about urolithin A. That's in the news a lot too. Pomegranates are a main source of urolithin A. So when you see all those advertisements for pomegranate juice, that's what they're talking about. So what do we know about urolithin A?
Yeah, quite a bit actually, and so that's sort of been up and coming, and we have a lot of interest in it here. Julie Anderson studies this from a neurodegenerative perspective, but Johan Auerbach and a number of other groups, he's in Switzerland, kind of did some of the early work that again show that urolithin A can induce autophagy, actually a more specific kind that's relevant also especially in muscle. And so a muscle, and like cardiac we talked about, have a lot of mitochondria. They have to generate a lot of force and use a lot of energy. So the mitochondria which generate all of this energy are especially important there. So urolithin A has been linked to, and it's called mitophagy. There are many different, it's sort of, it refers to what kind of selectric type of trash you're going to put into this bag. And so in this case, it seems like urolithin A is especially good at hyping up mitophagy. So turning over mitochondria that may not be working as well anymore, and therefore you can build newer and better ones. And so it's actually gone as far as the clinical trials where again, just because there are many things you can look at, and you should look at as many as you can, but the original ones have looked mostly, again, for muscle and sort of muscle output and strength and performance, and it seems to have beneficial effects there. Importantly so, in the studies, it was also looked at for autophagy markers, and autophagy markers were boosted in those situations, but again, correlation, we don't know if that's why you have the health effects, but certainly correlative, and a really exciting field. It's growing. It's soon to have like a conference on its own, just because there are more and more interest in researchers in that area.
Great. So let us talk about exercise, because I've gotten some questions about that too. Is there a connection between exercise and autophagy?
Absolutely, too, yes. So exercise is obviously something hopefully many of us can keep doing. Not everybody can exercise, but exercise has been linked to autophagy in terms of, again, boosting autophagy. In animals, again, model organisms, you can again do the experiment to ask if you need a full autophagy cycle to run in order to get benefits from exercise. That seems to be true. And so, yeah, this is sort of one of the most used paradigms in that regard. I love to exercise. You get many other benefits from it, right? You feel happy. There's a lot of enzymes and, like hormones that get triggered. Lots of benefits. Whether all of them are autophagy linked, I don't think we know. And maybe, that's also fine. We're not trying to say that autophagy is doing everything that may be good for a cell, but it's certainly helpful, including also in exercise. A key to exercise is that you also elevate temperature, and we're going to talk a little bit about that. So that can be another stress to the cell that work in good ways. And so a question could be, well, what if you did all of them together? Like, are you going to get extra benefits? We don't know that for all these purposes, but that's also something that's ongoing, right? Like if you both exercise and maybe dietary restrict yourself, you can get too much of a good thing, at least in principle. And so you kind of want to be careful with that. But those are definitely experiments that are worth doing, and we are.
So sleep, there's a note there that it's causal in flies. So sleep helps flies have better autophagy?
Yes, and it's not so far-fetched, right? Like, so sleep also has a period of quiet, and it's sort of, that's where you can clean up, right? And so there is a lot of indications, and this is a link to, many of you may know of these circadian rhythms, and so we have clocks and enzymes that help, sort of basically coordinate with the rhythm of the day. This can be a problem for like late night workers on shifts and things like that. That's not a good thing to change these cycles necessarily a lot. You can do sleep and flies. They do also live on a light, even in a lab, on a light and a dark cycle. And so you can basically look at sleep that way. And if you do some of these experiments where you can change some of these amounts of sleep, you can basically move these light and dark cycles. You will mess up some of the autophagy. So this idea that you can also cycle and that helps clean the single cells is incredibly important and a very interesting field to basically really understand which tissue is also, that's sort of a level that we haven't necessarily talked about, which each tissue may use different amounts of autophagy, like a muscle has to maybe do a lot because it works really hard, the brain too, right? It works very hard on a different kind of job, but all of them needs to stay healthy and well working.
So in case any of you are thinking about getting a sauna for the new year, this is your excuse to maybe go out and get one. So what do we know about temperature stress and how that might impact autophagy?
Well, there are good news about going to the sauna, independent of autophagy. So there is that, my Scandinavian neighbors of Finland, these are happy to go to the sauna. And there are again, correlative studies that would argue going to the sauna may have beneficial effects, for instance, on cardiac output, and maybe other effects as well. Neurodegeneration is something that's being looked at. But from a model organism's perspective, we do experiments where we can sort of hit the worms with a little bit of heat, and then you look at how well they do. It's what we call the hormetic adaptation. It's kind of, think like what doesn't kill you makes you stronger. And so if we have done this little trick of giving them a little bit of an exposure, like an hour, and we come back later to challenge them with a longer heat shock, they actually survive that much better. And we also found a few years back that in worms that are expressing like neurodegenerative, like clumping kind of proteins, neurodegenerative disease-like models, that they actually have lower levels of these aggregates, and that we could link directly to autophagy. So the idea is that the heat can opt the cleaning up process, right, building more trash bins and turning them around much better. You can clear some of that problem. We don't know anything about that in humans, of course, but the argument would be sort of the same. If you boost autophagy, and that's certainly true, elevated temperature in humans will increase autophagy. We don't know if that comes with benefits, again, correlation. But absolutely in congregate, good evidence that there could be a lot of positives for this.
So let's talk about stress for a minute, because there's so much like, oh, my stress is bad. Oh my God, you got to have less stress in your life, which in some sense is true. But there's positive stress and there's negative stress. So this is a positive stress, perhaps if it's done
right,
not too extreme.
Exactly. It has to be in full measure. Same for cold. There's a growing interest, right, in cold baths, and that's beneficial. Obviously, that has a lot of effects on hormones and other things. But it also, actually, in humans, there's evidence to suggest that also can trigger autophagy, or at least markers of autophagy. And so yes, there is a sweet spot. Probably not overdoing it, just like dietary restriction. If you starve, that's certainly not a good thing for you either, right? So it's finding that sweet spot, if you wish, and what's good for you and what's good for me may not be the same thing.
Perfect. We got a question about coldness, so we're on the same page with the audience. That's good. Okay. So what's needed to move the research forward?
A lot. We know a lot. So, like I said, all of these model organisms are absolutely instrumental in getting to where we are now today. We kind of know a lot of the players. We kind of know a lot of where things could be going wrong. We're knowing a lot about the types of autophagy. So we didn't talk about this today, but there's also something called lipophagy, which is turning over of lipids. So these little trash bags can be basically pulling in little pieces of fat and also help turn that over, which obviously can be super helpful helpful not only in metabolic context, but it's actually increasingly appreciated, and we have studies to that end as well, that lipid also affects something like neurodegeneration, which is maybe a little further fetched, but there's lipid toxicity that can basically affect neurodegeneration. And so we like to understand that mechanistically a lot better. So, when does this happen? Where does it happen? Why does autophagy matter? What is it it's putting in the trash bins? Is it the same kind? Is it different kinds? If we know the trash bin, maybe we can build more of that particular trash bin, and then help, in different contexts. A really important point is the second bullet points, and that is that we need better assays, meaning types of experiments to measure autophagy, and it kind of links to the next thing as well, and that is we need to get to humans as well, because as much as we love and really adore and also learn a lot from these model organisms, it will not replace what happens to you and me. So if and why autophagy works, who would it be beneficial for? Who would it make it sick? So, but in order to follow that in humans, we also need assays to kind of measure it, right? And so because of the process, and it's very dynamic, it's especially hard to measure it, because you can just kind of get a steady state. If you draw blood and you look at the markers, you get a steady state. You need to kind of measure how well is it running this cycle, right? And so it's a technical thing that just makes it extremely hard. And so you have to have biopsies, unless you, for instance, and that's what most work has been done in blood, right? And so blood, as you know, it may be sort of a window to the health of the organism or the human, but it doesn't report, equally well on all tissues, obviously. There are data to suggest that there are markers from in each tissue that actually can be found in the blood, which is certainly helpful, right? That means we can use blood to hopefully also learn something about other tissues. But we need ways to measure autophagy, for instance, in the kidney or in the heart to learn something.
So I think there'll be a quick answer to this question. Is there a simple, available test that an individual can take to measure their autophagy effectiveness?
No, not an easy available test, but that's the second bullet point, and it's absolutely imperative for the third one, which is this idea that for all the purposes of the supplements or whatever, we need to do what's called double-blind controlled experiments, right? Like in humans, where we can control for many of the other things that are important, like environment, disease histories, and so forth. And the plug-in to our, we have a clinical core unit now here at the Buck. One of the studies that are actually running there is a time trial where they're taking blood sort of round the clock. I would love if we could measure autophagy like that. That could become even better to these longitudinal studies, and we can better understand how health comes. It's a correlative study too, but you would have so many humans that there are many of these other factors that you can rule out as less important, basically.
So I got a question. Do changes in autophagy play a role in autoimmune diseases like arthritis? And somebody else asked, is there evidence that supporting autophagy could help protect joints or slow arthritis? And I know there's a difference between osteoarthritis and autoimmune arthritis, but do we know anything about joints and autoimmunity?
I'm going to say no at the top of my head, but in general, there are links between autoimmune diseases and autophagy. And so I wouldn't be surprised for arthritis as well. And I know we have other groups here, Birgit Schilling, interested in arthritis and bone health. That's something we would be really interested in looking at, again, by markers. It's possible the studies exist, I'm not aware of them. But I wanted to mention one other example of autoimmune diseases. So many of you would know the disease Crohn's disease, which is an intestinal disease. There are actually very clear and probably the best sort of molecular links and drivers are related directly to autophagy, something we're very interested in. Also trying to perhaps model in C. elegans that also has an intestine that can leak over time. So that you get older, the intestine tends to get leaky, and that's one of the culprits also of that disease.
Interesting. So here at the Buck, there's lots of collaborative experiments, projects going on. How about your lab? What kind of things are you working on with other folks here at the Buck?
In terms of with
Yes.
other groups, yeah. So so we're a relatively new group here. I came to the Buck like 4 years ago. Absolutely a reason to come here is many of these collaborations I had. So up till now, we've spent most time on sort of setting up our lab and getting other things going, but lots of interest in collaborating. And one of them we have started is actually with John Newman, who is head of the clinical core unit, where they are testing actively ketones. So you may know of these ketonic diets. And so these ketones have great effect. And we're now also, together with Gordon Lipko's group, putting them in worms, and they seem to be doing lots of interesting things, including actually improving this intestinal function. So we're really interested in sort of using this kind of collaborative idea of finding more mechanism in the simpler organism, and in that way they may have better ways to think about markers in their studies that are ongoing. So that's one example. And then with Julie Anderson, I already mentioned, who's interested in urolithin A and mitophagy. We're really interested in building bigger grants around that, including with Ashley Webb, who is a neuroscientist and very interested in brain aging. So we're trying to build around those ideas that the brain also uses a lot of mitophagy, that we can hopefully, from our perspective, coming from the worms with many of the sort of players, basically have ideas of what should these trash bin look like, and which kind of drugs can we kind of try that can maybe make even more trash bins, so we can turn over these mitophagy ideas better.
So at these seminars, I almost always get the question, how's your NIH funding going, and the activity that's happening at the national level? Is that how is that for you and your lab, and
Right.
how do you navigate that?
Yes, very important question. Obviously beyond our measures to control the NIH, but I would be lying if I'm not saying this is a very, very difficult time for everybody doing science, not just the people interested in autophagy for that matter. There are mechanisms, and I'm very grateful we have been and are supported by NIH for this work for reasons that obviously this is a mechanism that could be broadly applicable in many places, even if we don't study, a specific disease. So there is basic funding to be obtained from NIH, but the newer sort of, signs from NIH, obviously directed by the government, is that we also need to really prioritize human research more and down prioritize some of these model organisms. Maybe a little extremely put, but it's really important, and I want to make sure, and I hope everybody take home the message that there is a lot of good to be done with the simpler model organisms. And of course we need to go to humans, like I emphasized, so we kind of need to do it all, and I hope NIH will continue to appreciate that it's all connected, right? We're all trying to put pieces into the same puzzle, but it costs money. This costs a lot of money. It has aspiration and a lot of promise, so we need that money, or we need money from other places, basically, to continue to do this. But NIH absolutely has been the cornerstone in this.
Great. Thank you, Malene, for being here. Thank you for being
everybody.
here. Thank you, guys. I got great questions today. I'm so impressed that people are really into the biology of aging, so that's exciting. So, here's what comes next. You will get an email later today or tomorrow with the links to the video, to today's event, and also some links to some of the resources, the topics that we discussed today. I'm going to be back on February 4th, where we will take a deep dive into neurodegenerative diseases. I'll be talking to Dr. Lisa Ellerby. Lisa's a founding faculty here at the Buck. Her lab uses really cool technology to study brain diseases. She grows little tiny brain organoids from human cells and is doing some really interesting work with genetics and Alzheimer's. So that will be a fascinating event. I hope you're here for it. Clinical trials. Malene mentioned it. We have clinical trials going on here at the Buck. There will be folks out in the atrium afterwards if you want to talk to them about it. Who here is involved in one of our clinical trials?
Yay!
Yay, that's great. Thank you for doing that. Please talk to folks in the atrium if you are interested in learning more and are considering getting involved in our trials. How can you learn more about aging? So, we have an online course, Introduction to the Biology of Aging. We have a new course, Personalized Population Health. Both of these get really great reviews. Take them, impress your friends with your knowledge about how we age. Also, we have a podcast, and our season 3 is going to launch soon. We talk to some of the leading researchers around the country. And if you care about the science and you care about lifestyle, I recommend these podcasts to you. If you haven't been on a tour, please come on another one. We have them on Tuesdays and Thursday mornings. You can register for them on our website under our events. Keep up with us. We have a newsletter. We're on social media. We're out there as much as we can. You can sign up for our newsletter on our website. And finally, I would be remiss if I did not make a fundraising pitch. Everybody knows times are tough with the NIH. Philanthropy has always been important here at the Buck. It is now more important than ever. So any gift you give makes a difference here. Donations have been instrumental in kicking off our clinical research program. So please, if welcome your most generous self to the party before you leave today. Okay, folks, I think that is it. Thank you for coming. I will be back on February 4th. I think you should be here too if you can make it. Also, I understand there's sunshine out there today, so go out and get some exercise, boost your autophagy, and enjoy the sun while we have it. Thank you for being here.
Thank you, guys. That was awesome.